is the Eugene to Double Dutch pipeline demo, and I am Brian Jung, and I'm gonna demo for you today. This was a project for BE 552 in Boston University, synthetic biology, and I am in a group with Brian Mahabir, Landon Kushimi, and Zian Wang. So just to give you an introduction, our motivation was to create a way to perform both structural and functional specification. And so we decided to use both Double Dutch and Eugene, which are already tools that exist, and create a pipeline that a user can seamlessly go back and forth between the two tools, Eugene and Double Dutch, to optimize the final outputs. The technical challenge we had to overcome was making both Eugene and Double Dutch automated for our tool. So we wanted to find a way to assume factors without requiring user input. And to do this, we can actually utilize mini Eugene, which is a part of Eugene, and it's constrained that the user's input to assume factors and levels without actually asking for the user to input it again. And this runs completely on the back end. So to understand this a little, we have to talk a little bit about factors and levels in Double Dutch. So according to Dr. Rohner's paper, which talks about Double Dutch in detail, any component with a coding sequence is eligible for factor categorization. And in mini Eugene, any component that starts with a C are categorized as coding sequences. So we can assume that anytime a component starts with a C in the code and is included in counting constraints, such as contains, for example, the element it is referring to is a factor. So as you can see right here, these are all coding sequences. And basically what Double Dutch does is it finds the complementary level modules. So for example, if you have a coding sequence, it would look for a terminator and promoter pair. How we were able to assume factors without requiring users' inputs. So I was just explaining how we can actually parse the inputs for Double Dutch through the code from mini Eugene. And this is kind of a sneak peek of the function input parse that we used. So what this function does is it runs through every line of the code that was provided, and it looks for any keywords such as contains, more than, represses, induces, or drives, which gives us an example of the terminator promoter pairings or even just the factors. And from then we can look at if the first letter of the factor is a C, as you can see right here. And what that does is it tells us that is a coding sequence. A different part of the code for the second technical challenge that we have to find, and this was getting Eugene and Double Dutch to run in the backend. So this really emphasizes our automation aspect of the tool. And so what we wanted to do was create a function so that when the user presses a button, they don't have to input extra inputs into Double Dutch or have Double Dutch open up on a browser where they would manually have to plug everything in, because there would be no point. That would mean they can just use the two tools and do what we want them to do. And instead, so we made a function called open Double Dutch, and we use Selenium. And basically what that does is it actually takes the parsed inputs as well as other inputs that the user has already given us, and we assume their factors as well as what they want for cost function analysis, and we do that through the back end of Double Dutch. Entire workflow of our pipeline tool. So this is our GUI, the pipeline tool DD to Eugene cycle tool. So first what the user should do is find their CSV file. And what we mean by the CSV file is the DNA component library. So let me show you an example. The DNA component library Excel file should have promoters as well as terminators and their expression levels when they are paired together and to the corresponding cells. For example, when promoter 24 and terminator 12 are paired together, their expression is at the level of 14,500. So let's put that file in. Once you have that input, you want to give the level per factor, which means how many level modules you want each factor modules to have. So let's say 3. And for number of trials, you want to give how many trials you want Double Dutch to run to find the most cost-effective terminator and promoter pair in this example. So let's say 10. Finally, you pick the factorial design, and we are going to pick the full factorial, so any size. And finally, we want to put input a Eugene code. So this is the structural constraints. So I just had one that was written earlier. This is just an example. So it has the usual contains as well as more than exactly same count, as well as the positioning constraints and orientation, and finally the specification, which also would be important because our parse tool actually looks at this to pair the coding sequences with the promoters. So now when you press load, this is the new CSV file or the input that we would put in Double Dutch. It has that was everything that was already there, as well as the new parsed Eugene code, as well as their how they interact with each other. Finally, you press Double Dutch, and basically what that does is it's in the back end. It's actually running Double Dutch and doing 3 levels per factor module, as well as running 10 trials. And this is actually going to output 2 outputs that we are going to be using. Double Dutch has run. You look at the output, it says it gives you the cost of level matching, pathway homology, as well as DNA synthesis, and the total assignment cost of what you have given. But the most important part is that it will also output the library designs, which is, as you can see, we have inputted we said it contains C1, C2, and C reporter. These are the coding sequences. So the library design show us that coding sequence 1, coding sequence 2, and coding sequence of the reporters should have these terminator and promoter pairs, as well as the module assignment in detail, such as level 1 level targets. So these are the coding sequences, as well as the reporter, and it gives you everything, all the information you need. Open up Mini Eugene, which when you press the button, it opens up the web app, and you can actually have the Eugene code right here, press solve, and get all the outputs. But the most important part is right here, the solve button, where when you press it, right now it's not working because it's buggy. But basically what this will do is it takes all the outputs from both Double Dutch and Mini Eugene and do a find and replace, and it will basically tell the user what they have to do to get the best possible, most cost-efficient pathway design.